Yeah.
to see you. Nush, this is Søren.
Hi.
We used to work. Nice to meet you.
Long
Now-
time.
Ladies and gentlemen, welcome. Please take your seats. Please take your seats. Ellers, vi har jo sådan et horn. Ja, vi har et horn.
Ja,
det kan vi godt.
Lige ved at gætte.
Ja, ja. All right. I think we're about ready now. We just need the... Yeah. It is almost like herding cats, but are we about to have everybody seated? We guess so. All right, ladies and gentlemen, it's a great pleasure for me to welcome you here to the BioInnovation Institute to the very first day of Demo Day for the Acceleration Academy. It's a pleasure for me and for the entire team to see such a lot of excited people today. And we hope that today will bring a lot of enthusiasm, it will bring new collaborations, and it will bring a strong network and maybe also investments into our teams. The purpose of BII is to accelerate startups. It is to bring science out of the labs and into the economy to the benefit of people, patients, and the environment. The accelerator, the BAA or the Business Acceleration Academy, is the first important piece in that journey, where new and young startups can validate their business model and their commercial potential. And today we are proud to present to you the very first batch of teams who have been through our pilot run in the incubator or in the accelerator. Over the last 11 weeks, and I'm sure they will come back and tell you about it, but over these 11 weeks we have had them in our program. They have worked hard, and I tell you extremely hard. They have accelerated and they have achieved. And some of them actually achieved and accelerated more than what we thought they could. And I'm sure also that they accelerated and achieved more than what they thought they could themselves when they came here. Us at the BII, as well as the startup teams, are grateful for all the collaboration you guys have given us. It has been a concerted, ecosystem effort. I mean, the BII staff, modestly, we can talk about that, where we are 11 people and we have some expertise, but we have no way the capacity and skills needed to help the teams that we have had in our portfolio this time. So we are grateful and thankful for all the help that different mentors and experts and specialists have given to the teams during the journey here. So it's of very, very high quality what you're going to see today. And I am sure that you will find the pitches today of very high quality, both in terms of the ideas and the teams behind. I'm also certain that we will catch your interest, and maybe you will be interested to invest. It could be your money, it could be your time and your partnership, and it could also be both of that. But let's see, it's up to you at the end of the program. Today, we will also inaugurate what we have called the BII Acceleration Award. And that's an award that we are going to give out to the team at the end of the program here that we believe, actually that you believe, have accelerated the most over the BAA acceleration program. We will come back to how the voting will take place, but in like any American election, there will be probably some fraud votes and other things. But anyways, we will hand out the Acceleration Award at the end of the program. Together with the honor of receiving the Acceleration Award is also the right to come back at the next BAA Demo Day and hand over the prize to the next winner, but also you have the chance to tell about what happened to your project in the 6 to 8 months in between. So we keep track of the programs that accelerates from here. We also need help today. We need help to make the pitches and the discussions lively and relevant. So we have gathered a wonderful panel of experienced people from the startup environment. So starting with Sebastian Söderberg, Vice President from Novozymes of New Business Development. We have Søren Fransen, who is heading up Innotech, a tech transfer collaboration across Danish universities, and also director at Innovation and Research of University of Southern Denmark. And we have Stefan Kriskau, who is senior partner in Novo Holdings. So we have the entire, you could say, the entire pipeline here. We have the guys delivering the great science. We have the deal takers in form of corporates and investors. And with this, we are only minutes away from giving you the very first startups. I will hand it over to Monica, who will just tell us a little bit about the program. So, thank you for coming. I hope we'll have a great day together.
Welcome, everyone. This is a great pleasure to have so many great people in the room. Thank you so much for coming. Just to take you very briefly through what's going to happen today. First, we will have a mean and lean pitch session with our startup teams in front of the panel. Afterwards, we will ask you for your inputs regarding which of the teams have accelerated the most. After a short break, we will present you our incubation team, which we are proud to have already 2 in the house, soon to get more. And at the end, we will announce the winners. So, let the pitching session begin. First, I would like to welcome Mads Ebo Jensen, mentor of Binan Biotech, who will introduce the team.
Thank you very much. So today it's a great honor to introduce the Binan Biotech. I've been a mentor for the company for 3 months now, and it's been a great journey. And you have come really far, and I want to congratulate you on this. So what is Binan Biotech about? So Binan Biotech is going to develop a new novel treatment for osteoporosis, which is still a significant unmet medical need. And they are aiming at making a first-in-class, best-in-class treatment based on natural hormones. So everyone in this room understands that making a truly innovative medicine is hard. So it takes a good team and strong science. I think this company represents both. And one thing that makes the company stand out is actually not only have they come up with a new concept, but they have actually shown in people that it works. So they've done a human POC study. So with that, I hope you all are as excited as me. So please help me welcome Tue Imborg to the stage. Thank you.
Tak, Mads. Hello, everyone. My name is Tue Imborg. I am a medical doctor, and I am also the CEO of Binan Biotech. And today I'm here to tell you a little bit more about what Binan Biotech really is. Binan Biotech is a brand new Danish company and developing a brand new treatment to osteoporosis. Osteoporosis is a growing challenge, and it's a major challenge of growing old. And as we all get older in the world, the problem of osteoporosis is also growing. Today, more than 200 million people suffer from osteoporosis. Only 1/3 of people suffering are diagnosed, and of those diagnosed, only approximately half are receiving treatment. But osteoporosis is not only a problem for the elderly. It's also a big problem for a small group of young women. It's a big problem for a group of young women that want to have kids. These include women with anorexia. These include women on glucocorticoid treatments. These include women with autoimmune diseases. And the reason why this is a huge challenge for them is that the treatments on the market today are not really working for these young women. The bisphosphonates that are widely used are teratogenic, so when these young women want to have kids, it's not really a good option. The 2 other major treatments on the market, Forteo and Prolia, have heavy rebound effects when they are not used together with bisphosphonates. So for these young women and many other patient groups, Binan Biotech might be able to make a real change. So how exactly is it that Binan Biotech is making this change? So Binan Biotech is following a quite nature-true approach to treating osteoporosis. We are following the natural response of the body to eating, and we are doing that by simulating or stimulating the gut hormones, intestinal hormones. In a lot of ways, our approach to treating osteoporosis is similar to the approach that Novo Nordisk took when developing Victoza using GLP-1. So Novo Nordisk used GLP-1 to develop Victoza, whereas BioInnovation Biotech is building our treatment on their sister hormone, GLP-2, and their common cousin, GIP. So cutting it very simple, what we do is that we take the effects of these two hormones and put it into one product. And why do we do this? We do this because we know that it works. Like Matt said, we know that it works and we have tested it in humans. We have human trials on healthy people showing that GIP as well as GLP-2 have pronounced effects on the resorption of bones. We also had trials showing that when you combine the two, it has an even more profound effect. We're also doing this because we know that we can do this. We have already developed a portfolio of compounds that have the effects of both GLP-2 and GIP. We call these dual agonists. Some of these dual agonists we have actually managed to produce so that they have, relative to the natural hormone, 100% the activity on the GLP-2 receptor and 100% the activity of the GIP receptor. These could already today be potential lead compounds, but we intend to make them even better. So I'll use the last couple of minutes to explain a little bit more about where BioInnovation Biotech comes from. We are a spin-off out of University of Copenhagen, and the whole idea started back in 2016. Since then, we have developed compounds, we have filed patents for the compounds, we have initiated human studies testing our way of treating osteoporosis, and had the first results in summer 2017. Since then, we have spread out these studies to other patient populations and other groups. Then in 2018, we had the luck of being invited into BII as the first generation of Genipex here, which really helped us accelerate the business. We got the opportunity to work with a lot of very competent people on a lot of issues that normally would be left alone in the small biotechs with young people like us. We had the opportunity to work with Plesner and Masanti to found a company on the real terms, right terms, getting it legally ready. We had the opportunity to work with accelerators on doing a business plan, doing market analysis, also working on how to pitch. We had the chance to go to BioEurope, get connected with a lot of potential collaborators, get connected with a lot of potential investors, and getting connected to a lot of people in similar situations to help us move on. We also had the opportunity to work on the company Visual Identity, and we had opportunity to work with the IP together with IP patent lawyers, and really this has been a huge benefit for a company like us in such a complex industry as the pharma. I would like to use the last couple of minutes to just introduce our team. So we have a quite science-heavy team. We have experts within intestinal hormones. We have experts within bone metabolism, and we have experts within drug discovery. And behind these, and when I mean expert, I'm talking Jens and Bolette and Mette, who are sitting out here, all of them. Behind these experts, we have a pipeline of young PhD students who have already been doing the studies so far, so they know how to do it, they have done it before, and they are ready to jump on board when we get funding. On the business side, we have a couple of young business developers, and we are trying to build a strong and active board around us, and this might be a place where we could need your help. So at the moment, we have a company that is almost ready to go. We need the financing, and this is what we're going to spend the winter looking for. And I hope you'll support us in that. Anyways, thanks for listening.
Thank you very much, Tue. Now we would like to hear some comments from our panel. Christian?
Yeah, so thank you for this review, and of course I appreciate it's a very short pitch, 7 minutes is not a lot. But going back to the slide on the biology, you showed effects, short-term effects on bone turnover, And this are going to be a treatment for the long term, for sort of chronic treatments. So what do you have to support that your effect will actually have a long-lasting effect rather than just a transient effect?
So there's a couple of things. First of all, we don't have the long-term human studies yet. Of course, these kind of studies cost a lot of money. We have the fact that we are somehow stimulating quite high up in the mechanism. So a lot of the other treatments that we see, Prolia and Forteo, they target a specific small molecule that they kind of block with an antibody or these kind of things. We are somehow stimulating a quite high up response that the body has to eating. And it signals this through these two hormones, and then it probably has a lot of cascades that kind of, you know, are second messengers to these kind of systems. So stimulating up high kind of takes some of the risk out of very quick sensibilization by the body, I think. But we really need longer studies to be sure that it also works in the long time, and that we don't see the same rebound effects that you see with some of the other treatments for osteoporosis. Definitely.
And then a follow-on question, appreciating that these hormones that you're working with are normal physiological hormones, and they actually do more than just work on bone. So are you concerned for some of the non-bone effects of the hormone treatment?
So one question that we have been asked a lot is about the GIP and their effects on obesity, and whether that might be an issue for some of these patients with osteoporosis. Especially the elderly, frail, thin young women, which are kind of like the archetype of a lady with osteoporosis, this is not going to be an issue. In general, these hormones are quite side effect mild. Also in our human studies, we have not seen any sort of discomfort with the patients, these kind of things. Yeah, I think that's the best answer I can come with on that.
I think you did a great job in explaining exactly what it was you were trying to solve here. I like the references to the amount of patients. I like the reference to Novo Nordisk and their approach. What I maybe lacked was to understand exactly what kind of funding you need by when. When are your next milestones? What's the critical things that you need to address now?
Okay. So the next round of money we need is to further develop our compounds. We would like to work more on that, get to the lead compound that we can take into a preclinical. We think we can do that quite quickly. Within a year and a half, we should be able to have a lead compound and take it to preclinical testing. That would amount to $10 million, these kind of monies, $5 to $10 million, over the next 2 to 3 years. Yeah. That is the amount we're looking at. Yeah.
Thank you very much. Due.
You're welcome.
And now I would like to welcome on stage Thomas Kledal, who will introduce next team in Proter.
Thank you very much. I have been a coach for Improter in this process here. Where are they? I don't know where they are. There, Peter and Nikolai. But really it's been a very, very easy job because they are so self-driven. With Peter's high-level science and Nikolai's business background, it's actually been quite an easy task. And I hope it works out as you will see. Improter is really all about making cancers visible to the immune system. Hence, you know, going directly into the immuno-oncology hype that is really spot on right now. Peter and I goes a long way back. So I think we've known each other for basically 20 years, published our first paper together 18 years ago or something like that. And back then you were a young, rebellious scientist. Now you are experienced in successful technology transfer and going with IP all the way to actually clinical studies with big pharmaceutical companies. You have done that. And it all starts with, you know, really fascinating science on targeting and retrovirally encoded DNA elements, but now actually showing how they work in tumor models in mice. And building this fantastic company with a focused approach to, for instance, breast cancer and head and neck cancer. I just want to say one last thing, is that when I look at you now, you are still a young and rebellious scientist, and I hope you will stay that way no matter how successful Implantea is going to be. So please welcome Peter on stage.
I think yes. Am I on? Yes. Thank you, Thomas. That was a good introduction. Yes, my name is Peter Holst. I have been doing vaccine research for the last 15 years, and my research and my team's research has entered into Implantea now, where we are trying to work with immunotherapy. So immunotherapy is a very exciting discipline within science, because we now know that it's possible to cure even late-stage cancers. Immunotherapy can currently cure a few cancers, a tiny bit of the total spectrum of cancer. But we can see rationales that maybe many more could be cured in the future. However, the majority lies out of reach of those treatment principles currently being advanced. So in order to get to those, we need to understand what kind of tricks are cancers using to hide from the immune system. Now, the trick that we have discovered in old literature has basically come from our ancestors. Monkeys, early humanoids, were infected by retroviruses. These retroviruses stayed in our genome, and now 8% of the genome of modern humans consist of ancient retroviral DNA. These elements are necessary for cancer development and always overexpressed in cancers, with a few exceptions of virus-induced cancers. Now, what's this connection then? So the immune system obviously is designed to kill viruses. So the viral infection of a host cell has in common the introduction of a viral genome into the host cell, and that introduction is a powerful immune stimulus. Any successful virus must have found a way to counteract this. And the trick that retroviruses use is actually to have a patch, a sequence within their attachment protein that binds to host cells and injects its nucleus or its nucleotides. That patch is inhibiting the immune response towards the viral genome. Now, we take this fusion protein and we mutate the immunosuppressive patch and insert this into a clinically proven, very potent immunostimulatory vaccine vector. And the result is a potent vaccine that can direct a powerful T cell and antibody, basically the cellular and the humoral immune response towards the cancers. Once we have done that, cancers are now targeted by their own immune evasion mechanism, and now they're vulnerable. Now, this is probably the only primary data I'm going to show, but what you see here is basically an example of a therapeutic trial against colorectal cancer, where the animals that are being vaccinated in the black are rapidly clearing the cancers, controls are growing. And what we did to prove the generality of this approach, I think this is good enough really, but to prove the generality, we took months later and challenged these mice, not with colorectal cancer again, but with the breast cancer in the mammary glands. None of the animals could engraft a new breast cancer. So this is a curative and prophylactic tumor immunity. Now, the potential of such an approach is obviously enormous. More than 1 billion people today will die from cancer, at least unless we do something about it. Now, we're not there. We are actually at a stage where we're trying to get into human trials and prove our point in humans. But with a general approach, there are many options for us. We can see that other companies who have succeeded in generating early-stage clinical data have made either billion or multi-billion-dollar evaluations and exits. And this is what we are trying to get into. Now, we went out with this data package to the scientific community in Denmark, in Europe, in the US, and the response was uniform. They all loved this data package. And actually, all of them wanted to be part of it. Now I'm showing you a quote from Jay Pesovsky, the head of the vaccine branch of the NCI, in the summary stating that, could I play a part in this? And this is the feedback we get. Now, with the support of the BAA and this kind of feedback and this kind of support from the community, we've been able to establish a good team to take this forward. Now, I am the only person actually in the world that has succeeded in making better immune responses to viral vectors as they are. I can increase the response of viral vector vaccination and have that track record. Nikolaj Høyer Nielsen is a serial entrepreneur and a guy who really can take companies up and get them moving. And he's been doing this, and he will continue to do this in the future. We have been able to recruit Anne-Marie Anderson from Oxford. She has a background in having developed this core platform technology that we're going to use, and it's a perfect profile for our platform development. Future employees, we can recruit a really experienced CEO who has field-specific business development experience for many, many years, hundreds of deals to our company. So we have the business platform in place. And we can also recruit the animal model specialist and the oncology trained staff we need to go forward. We have access to supports in biotech companies specialized in vector development. And for our later development, we have access to Inge-Marie Svane and Per Thor Straten at the Center for Cancer Immunotherapy, which of course said, can we run your trials? So that support is there as well. Now, if you look at our progression in the BA, yeah, this is a stereotype, but I think it holds true. We had basically HIV and cancer and whatever on the palette a few months ago. And now we have a focused approach to target cancer and get this into humans. Our approach has been validated by Big Pharma. We got positive feedback from investors. We have freedom to operate. We own our own IPR. We can assemble the team. What we need now is the research infrastructure and the financial support. And then we can make rapid advance towards clinical trials and basically into the market. Thank you.
Thank you. Thank you, Peter. Do we have any comments from the panel?
Well, thank you, Peter. Very good presentation. Obviously, I don't know how many presentations you've given to me over the years, but it's certainly a lot. And I do appreciate that how the focus that has been brought into your project, as the mad scientists there indicate, there's always been a lot of ideas. So great, you are now concentrated on the immuno-oncology approach. My question then is, given that immuno-oncology is a very crowded space, a lot is happening there. There's more than 1,000 combination trials currently going on with all kinds of therapies. How do you see this approach fitting into the clinical practice?
I'm so glad you asked that because I was saving some time and didn't explain it myself. So basically, there's a fantastic hub now in targeting cancer-specific mutations. This is the majority of the hub. We don't do that. We target a shared fundamental principle of cancer development. And we're basically separating us from the vast majority of this hub. Now, they can only target basically in very few cancers that are not smoke and sunlight induced. They need mutations to do that effectively. And we are aiming much broader than that. I can also say that we have reproduced past failures. So I found the first immunotherapy trial using virus sequences back in '81. If we make this kind of approach without introducing our prime mutations, we cannot direct immune responses against these viral genes. They are immunosuppressive. So we have basically proven the difficulties, but also solved them. And I guess this difficulty in getting a response up may explain why cancers have stayed vulnerable to endogenous retroviruses, even in their evolution. I think we stand apart by targeting a different segment of cancers, by very, very good preclinical data that I think most would be envious about. And then this broad cancer approach that we basically can find our best position. If we get sufficient funding in, we have a platform technology now that targets functions that we can see are essential for cancer immunization. Unlike other checkpoint inhibitors right now, we're actually talking about cancer-specific immune checkpoints. So I think the differentiation is strong enough. What we need now is more support to build a stronger data package and develop the platform.
I appreciate this is sort of the first step of an answer, but I do also think that the world in oncology is super complicated and that there are other projects doing similar or at least comparable things. It's not all of the projects. But all right. But what would you? What is your current funding requirement? What are the next sort of key milestones for this project? Where would you like to see sort of the next critical experiments go?
Right. I mean, we will be tremendously happy to enter the BI format here because we have a few things to address. We would like to go into clinical trials with the ability to stratify patients based on our target selections as a one key thing. So right now, we'll be happy to raise the next 10 million kroner from BI and maybe some extra funding. But very rapidly, we'll be looking for the funding required for our clinical data package. And we have a hurdle. We're using viral vectors. They're expensive tools. And we need 3 million just for the GMP and the tox. So we're probably talking about 10 million euro after this phase, which will start hopefully after getting some money from the BI.
And then thank you also from me. Excellent presentation.
Thanks.
Very short. What is it that makes you sleepless during night? What is the major challenging you're facing right now? Is it finance, infrastructure, or is it on the science part?
Well, I have no funding next year. So I have basically stepped all the way into this. It is not true. I have 20% at KU next year, basically to have an easy access to animal facility. So I'm gambling all on this, and the deadlines are getting close. So that is an issue, of course. I think we're on a good track. I hope you all agree, and I hope that the BI agrees, and then we're fine, and I shouldn't lose any sleep. But from that, we're talking about a new class of immunotherapy, and I really want to see this work in humans. I mean, every clinician we talk to say this is data package deserves to be in human trials, and obviously until we are there, it may not work.
Thank you very much, Peter. Thank you. With this inspiring note, we will move to the next mentor who will introduce Circle Biomedical. Welcome, Finn Kettler.
Thank you very much. I said I would like to introduce Circle Biomedical. It's a company that has gained a lot of attention lately. They are supported or backed by Melinda and Bill Gates Foundation, and they have also had some interest from different investors. So it's a company that you should really look after. Circle Biomedical is a company involved in birth control. Not condoms, not contraception pills, but a small capsule sold as a capsule and placed in the vagina to perform a barrier that will prevent the sperm getting to the eggs. It's a new hormone-free product. It is on demand, it's without side effects, and requested by millions of women. Please welcome Thomas and Frederick.
Thank you. We are Circle Biomedical, a mucus engineering company developing a novel non-hormonal contraceptive for women. Today, there are 125 million women that use a contraceptive in Europe and in the US, on average for more than 30 years. For more than 50 years, the number one solution has been the hormonal contraceptives. However, this is changing, and it's rapidly changing. Due to increased awareness of the side effects and also increased scientific evidence of the side effects. Today, we, for instance, know that the risk of developing a depression is increased by 80% when using hormonal contraception. This means that today, more than 28 million women believe it's extremely important that their contraceptive does not contain hormones. The problem is, if we look at the market, the alternatives are extremely limited. So the products that are convenient to use are highly invasive, either being a systemic drug or an implant. On the other hand, the products that are not invasive are inconvenient to use. So the technology we are bringing to the market fits this gap in the market. And the value proposition that we're bringing is a minimally invasive approach to contraception, where we only modify the mucus layer in the cervix. It's highly convenient, working within minutes, having extended protection, and we focus on creating a great user experience.
Right. So the way we'll achieve this kind of unique value proposition is by leveraging the woman's own natural barrier. And in the reproductive tract, that is the cervical mucus. So it's a gel that sits between the vagina and the uterus. There's a cervical canal. And that gel is a really good barrier to sperm cells, actually, throughout the menstrual cycle. But then around ovulation, it loosens up completely and then allows sperm cells to go through. So what we're doing is we're delivering these capsules that you can see here that dissolves in the vagina and releases the polymer that can interact strongly with cervical mucus. And by interacting, it closes down the pores and prevents the sperm from going in. Then the sperm are maintained into the vagina, and where local, like acidic pH will deactivate them pretty quickly. Now, the effect is reversible, since natural turnover of mucus will shed kind of that complex away after some time. Now, we have very strong in vitro proof of concept. Here you see two images, one to the left where sperm can happily swim through unmodified mucus, and to the right we've modified the mucus and it forms this kind of capsule that sperm can bump their head against. We also have access to human ovulatory cervical mucus and human sperm, and we developed these assays where we kind of recreate an artificial cervix filled with cervical mucus that we expose to sperm. and then look at how sperm swim through the capillary tubes. And you can see the results of the experiment. We've done a number of times. You can see untreated mucus with a distribution of sperm along the capillary tube. And when we treat the mucus, that completely blocks the entrance of sperm. So we have 100% efficacy in vitro. The polymer that we're using also has a very strong track record of safety. We've done assays on using in vitro models of epithelial cells showing no cytotoxicity there, and also on sperm, showing through these DNA fragmentation data that there is no cytotoxicity on sperm cells. We're just targeting the cervical mucus. Now, the results, scientific result, and our scientific approach in general has been validated really by major actors in the contraceptive space. So the Reproductive Health Investors Alliance has gone through a very thorough 4-month due diligence process and is now actually our first investor. And there's also a big pharma company that we've been discussing with, have reviewed it with internal scientists, and have provided us a very strong letter of support as well. Now, we have a patent that covers this group of polymers that can achieve very efficiently this barrier reinforcing effect, filed in 2017, and it is owned now by Circle Biomedical. In terms of regulatory path to market, we're classified as a Class 2B in EU and Class 2 in the US, so a medical device, not a drug. And we're very fortunate to have this FDA guideline for vaginal barrier contraceptive that we can follow. And it's composed of these 2 phases comprising 250 subjects altogether. And in the next few months, we'll finalize our classification by leveraging these 513G mechanism in the US and contacting also in EU the notified bodies.
Yeah, so we have exciting opportunities in terms of go-to-market. We can partner with one of the established companies in the industry, but we can also use some of the new online platforms and thereby access millions of users from day one. But we could also create our own platform and control distribution. We're focused on building a team of A players from academia and from the medtech industry. I'm Fredrik Madsen, I'm the CEO of Circle Biomedical. I've been working in the contraceptive space for the past 4 years, developing an in-depth understanding of the user needs and the commercialization process. I've also been working in the Danish MedTech Association, building a strong network within the industry.
Yeah, and I'm Thomas Jose, so I'm the CSO of Circle Biomedical, and I'm also a researcher and group leader at the Royal Institute of Technology, KTH, as in Sweden, in Stockholm. And I have a background in bioengineering, and in 2011 I went to MIT for a postdoc in a group working on mucus. And so I kind of fused my background in bioengineering and started seeing this mucus as a material that we can actually engineer via topical treatments. And so that led to this mucus engineering field that we're working on. And we, about 2 years ago, I met Fredrik Madsen, and we, by talking, we realized some of the inventions that we're coming up with in our group can be translated into really new technology for contraception.
Yeah. Our next key hire is the CEO to bringing experience transforming an early concept into an approved product. So during the BAA program, we've matured our case in a number of ways, regulatory affairs, IP, product design. We've been able to validate investor interest from some of the later stage investors. We have raised pre-seed funding from the Reproductive Health Investors Alliance in San Francisco. So really from entering the program, we came as a team with a good idea, and we leave as a funded startup. Thank you.
Sebastian, do you have comments?
Yes, yes I do. Thanks again for a great presentation, and I think you have a very clear aspiration. I'm not sure I entirely understood your go-to-market approach. You say you had have three different opportunities, but I guess you could already bring them in now to de-risk your clinical studies and make them part of this journey. What are your thoughts around that?
Yeah, so we already have discussions with one industrial partner. They're very interested, but we want to keep our options open because we don't think necessarily it's the best path to partner up with one of these companies. We also had a meeting with one of these online telemedicine solutions, trying to discuss if they could be interested in partnering up, and they're very interested. So really we see these options, and we also see the option of going alone with the right kind of funding. So at this point, we're still keeping all options open.
Yeah, I also want to thank you for a very good presentation and very clear sort of the concept. So obviously, eventually you'd have to do a larger clinical trial, but what is on the near term sort of a major de-risking activity where you know that the observations you've done in vitro in a petri dish somehow is representative of what happened in real life? Obviously, there are a few steps between the petri dish and and the actual life. So, what can you do in the short term to demonstrate the effect that you need?
Yeah, in terms of preclinical studies, we've divided that actually into 2 phases. But the first phase is really to do exactly that, de-risk as fast as possible. And to do that, we'll go in an animal model. So we've identified sheep actually as a proper one. And we'll use kind of an innovative technique. We could do live imaging by using fiber optic confocal fluorescence microscopy. So basically, we can track the presence of these polymers, but also the presence of the sperm within the reproductive tract. So we can understand where the polymer interacts, how long it stays there, what are the dynamics, and then we could track the sperm localization as well, making sure that we do have a barrier in there. And that should already kind of comfort everyone that this is feasible in vivo.
And how much money do you need to come to that point?
That's 0.5 million euros overall with the other activities. Yeah.
Thank you very much.
Yep, thank you.
Very interesting presentation. Now I would like to invite to the stage Tom Olsen, who has been mentoring HeartView Medical.
Thank you very much. It's a great honor, a pleasure to introduce you HeartView Medical. We all know about the heart. It's the most important muscle in the body, the engine. If that engine fails, we are in trouble. We all know that. HeartView have developed, based on a very, very strong IP technology from both US and here in Denmark, a new technology that will give us more detailing, more dynamic view in 3D of the heart. That will enable us to make much more precise diagnostic of this engine. And in that view, it's been a very great pleasure to be mentor and listen carefully to this great team. Thank you.
Thank you very much, Tom Olsen. Today, we do not know exactly how the heart is working. We have patients that are being diagnosed in a non-optimal way, which means again that the therapy chosen is non-optimal. We can change that. With our technology, we have shown things that has never been seen before in the heart. We can change the way that cardiology is taught today. We are HeartView Medical. My name is Marian Andersen. I'm CEO, and we have our data scientist, Martin Vandborg Andersen. Welcome. Apart from myself and Martin as part of the team, we have 2 hardware engineers based in the US. We have a collaboration between Aalborg University and Duke University. Additionally, we have 3 experts within the field of echo to support us, and they're also part of the company. In the middle, you see Peter Søgaard, who is a cardiologist, and then we have Samuel Smith, who is our data scientist expert from Aalborg. And to the left, we have Olaf van Ram, who is one of maybe 3 persons in the whole world who is able to define the technology we are working in as of now. Most of us have experience from the startup entrepreneurial world. Many patients will be able to benefit from this invention. First of all, children, because children have a very high heart rate that is difficult to follow today with existing technologies. Secondly, we know that populations are growing older and older in the future. It's always a challenge to society. Even today, 10% of people above the age of 65 develop heart failure. And that number is going to increase. We'll be able to diagnose these patients earlier, meaning a better therapy can be chosen at an earlier stage. Where our focus will be in the beginning will be those patients who have had a cardiac resynchronization therapy device implanted. It actually turns out, even today, that 30% of these patients do not benefit from the implant. So it's a waste of money, and it's not very good for the patients. Today, nobody with existing technologies can show what the reason for this failure rate is, but we can show it.
And the way of doing this is by high frame rate three-dimensional ultrasound, which will be the ultrasound of the future. If you take a look at this picture here, you see a three-dimensional image of the heart. And in red, we have colored in what the views you will get with a two-dimensional ultrasound image. Everything outside that red area you can't see with ultrasound today. And everything in yellow is the areas that are actually interacting when you actually have a contraction, which is a huge problem because you simply can't see it. Not only that, if you look at the speed of ultrasound, what you can see here is a golf ball, of course. And you can see that it's moving, but not really anything else. But if we increase the speed of the image, all of a sudden you get these beautiful details about what is going on with this golf ball, what is happening to it, how it's deforming in time. The same goes for a heart. If you record it at a very high frame rate in 3 dimensions, you get these beautiful pictures of how the heart is deforming as a function of time. And that will be a great tool for doctors to use in the future. Not only that, if you look at an ultrasound image in general, they have fairly poor image quality, which means that in many cases it can be difficult for a doctor to actually find out where exactly in this image the heart is and where there's the blood. One way of solving this is by using a contrast agent, which is an invasive method where you put microbubbles in the bloodstream and thereby emphasizing the contrast between blood and myocardial walls. But that's invasive. What if you could do that in software, like ours? All of a sudden you get the tools for the contrast improvement without that invasive methods. And it gets really easy to find out where exactly in the picture, where the myocardial wall is. Again, a huge benefit for society in general. So tomorrow, what you will see from us is a high-end 3-dimensional ultrasound system recording at roughly 500 images a second in 3 dimensions to get this 3-dimensional image of the heart contraction. Together with this, we'll have software that goes in and uses AI support for both acquisition and also the automated analysis afterwards. For diagnostic purpose, this means that we'll reduce the need for contrast agents for the patients, which in turn will lead to less need for MRI and CT, which will be referred from the echo department. And you free up a huge amount of area that you can use these machines for other patients who will need it. Not only that, if we go back to our CRT candidates, every single year in the US and here in Europe, 50,000 patients get an implant that won't help them at all. 50,000 people. Now imagine if you didn't have to give them that implant.
So our strategy to get into the market will be a 3-step strategy. First step is to sell software packages to tier 2 ultrasound companies. Second step is to work together with hospital cardiology research institutions so that they can get started to find good patient cases that we will use further on. And third step is to go with the volume market, where we will have to find a strategic partner to work with that has the sales network that we need. So first, after 2 years, sell software to existing companies like Canon, Esaote, and before we go out to the volume market, we are looking for partners like Siemens, Medtronic, who are doing something in cardiology but do not have ultrasound. The progression that we've been through over the past 3 months has been tremendous, and seeing is treating. So if you can't see it, you can't treat it. That's the main purpose of us being here. Heartview Medical. We can change the way you treat patients with cardiac diseases. We'll be very happy to introduce you to our new web page that we just launched today, and to see us at you at our booth. Thank you very much.
Thank you. Do we have any comments?
So thank you. It wasn't absolutely clear, but I assume you use standard ultrasound equipment, but your technology is an algorithm that treats the image. Is that correct?
Yes and no.
Okay.
Initially, we'll develop a software system that will be able to be used with conventional ultrasound systems. And that will be our first approach to getting to the market. Afterwards, and this will be in 2 years where we'll start to get the initial revenue, then in a few years from now, we'll have a fully evolved three-dimensional high-speed ultrasound system that simply isn't existing today that we'll be able to use. And we'll also produce that as well, yes.
So you would both develop your algorithm and validate in clinical trials that algorithm, as well as developing a brand new ultrasound machine. That sounds like a quite ambitious undertaking. How much do you need? How much investment do you need to achieve that?
We need $30 million in order to get to that point, and we've divided this into 3 different milestones. First one is to have the software solution ready, which will cost a bit more than $2 million, and then another $8 million to develop the prototype, so we're ready to go out and do clinical studies in different parts of the world, and then another $20 million to develop the fully hardware system with the integrated software.
Okay.
And this will happen over 5 years from start.
And did you, in those $30 million, put any funds for commercialization to set up sales, and that all
yes,
requires
we did.
investment?
But only sales when it comes to research institutions, because we know that there are other bigger players out in the market, and we're not going as a startup company to set up a global sales network. So at that point in time, we will find a partner like Medtronic, who is well known for doing acquisitions within this area, who have a worldwide sales force, who are in cardiology but do not have ultrasound. That will be our strategy.
And given the time, thank you for a nice presentation, easy to understand, and I'm of course inspired by the efficiency into treatment like the ones you presented here. Any thoughts on that this type of technology could actually be used in other industries? I mean, I could see a tremendous potential in other industries in terms of actually viewing and seeing. So maybe it's a completely different market in 5 years.
You're absolutely right.
Any thoughts on that?
Yeah, absolutely right. Because when the X-ray was developed once, that person, Mr. X-ray, what was he called? He did not know that actually we're using the same technology to scan suitcases in the airport now, right? So we know, of course, you can use this also for other organs in your body. The only thing is the heart is the one moving most fast, right? So, but in other industries as well. It's a completely new technology, so yeah. Thank you.
Thank you very much. Now I would like to welcome Newt Goldsmith, who will introduce 5th startup firm BioTX.
So hello everyone, and as mentioned, it's my pleasure to introduce Firm BioTX. Firm BioTX is maybe thought of as on the one hand, taking the industrial microbiology experience of Novozymes and the animal health experience of fermentation experts, and marrying them to create a consumer-facing company that uses the microbiome and the benefits of dark green vegetables to create products that benefit consumer health and the health of their pets. It has an awful lot of animal data. It's entering human clinical studies, and somewhat I think it's one of only 2 companies that actually has real customers and real sales, from which you learn a lot. So it's my pleasure to introduce what I hope will be the Danish equivalent to BioGaia.
Thank you, Neil, for the introduction. So my name is Søren Kjærulff, and I will talk about Firm Biotics. So Firm Biotics is a BioInnovation company trying to help and support, improve the gut health of pet animals, but also of humans. So the team consists of Tina, she's here, and she's responsible for the marketing and sales. She has very good experience from the mother company, Fermentation Experts, selling fermented products. And then it's me. I have a background from Novozymes, working together with Sebastian. But most of the time I was heading up the biopharma R&D activities in Novozymes. So our mission in Firm Biotics is to help solving the problem of worldwide increase of metabolic and also autoimmune diseases that we see in the Western world, but also now in China and so on. And our solution to that is this fermented seaweed and plant product. It consists of prebiotic fibers, of probiotic bacteria that we use for the fermentation process, but then of a lot of different bioactive molecules which have different activities. So what we can with this product is that we change the bacteria, the gut bacteria, and in that way we prevent inflammation, for example in the guts, but also other chronic related inflammations. So the last 2 years we have spent, 1.5 years, we have spent a lot of time developing these products, so we have a very good process now. We have also tried to understand how this complex product actually works, and it is really complex. And as you just can see here, we have investigated all the metabolites in these products. So here on the top you see more than 400 different compounds. Some of these compounds, they are antimicrobials, others are antioxidant, and again others which we have isolated and tested in different cell models, they are anti-inflammatory. But if you look down here, we have, as Neil also indicated, carry out a lot of animal studies. We have also created or tested a lot of different human patients. But when you see this slide here, we can actually modify the microbiome of pig gut bacteria. And what you see here, the first one, you can see just pigs receiving normal basal diet. We don't see a great diversity. We see a lot of the gram-negative bacteria which are responsible for the chronic inflammation. Whereas when we see on the results here, on the left side you can see the pigs which receive 2.5% of our fermented product. It's much more diverse. The composition of the gut bacteria is well distributed. We have decreased the gram-negative bacteria, but we have increased the good guys, the lactobacillus in the microbiome of these pigs. So based on this research that we have carried out, we have just started a clinical trial at Silkeborg Hospital in Crohn's disease patients. And we will receive the data from this in a year. They have actually already tested the fermented product in a lot of patients and seen very good results there. So we are looking forward to these results. So our business model, our focus is that we are looking into the pet food supplement area, because a lot of the things you see for humans is also the same issue with pets, the way we treat it today. We give them too much to eat and too much fat and so on. And then we are also then looking into the food supplement area. Together with the BII team, we have looked into a digital platform. And we have just decided that we actually also want to sell from business to consumers. So we have just introduced an online shopping website, as you will see a little later. But then we also focus on business to business. And in that area, we have very good leads on customers and partners in Poland and Germany, which we are following right now. So here you see the example of the packaging material for the pet shape product, which are for pets. And this was developed together with the team Christian, which are here, and a commercial company. So we will actually introduce this in a couple of days. It's ready, so we can actually start selling in this area. As I said, we have also realized sales within racing horses. So we have 3 customers now in Denmark which are buying this for horses which have got problems, and a lot of horses have that actually. We have made a digital platform, and here you can see some of the signal mining that we are doing on the internet right now. So for the food area, we have chosen to look into the lifestyle area, but also the health and the sports segments where we believe we can sell our products. So we are finalizing this right now, and then beginning of next year we can introduce it. So here's the milestones of Firmbiotics the next year. It looks like a roller coaster, and I'm sure it will be. That's how it is. Creating business, but we are pretty sure that we will succeed with this plan. So we have introduced, or we have started the clinical trials in Crohn's disease patients together with Silkeborg. We have just set up this pet food shop, and early next year we will still also introduce our product for food. We have been discussing a lot with the experts if we should do both, but we'll still do that. Then end of next year we'll get the data from our clinical trial at Silkeborg, and of course we are looking forward to that. We have actually received money from a EU grant to start up a clinical study in diabetes. So we'll do that also. And then, of course, we will also need to earn some money. So we are looking for customers. So if you want to buy the product, I'm pretty sure you will get a lot of help using it. But we're also looking for competences for our sales, but also improving our process and get it more robust. We would like to have that. Okay, good.
Thank you.
Yeah.
Søren, first of all, congratulations with the speed that you have established the company, and you got the first commercial partners on board. I think that's very impressive.
Thank you.
Having said that, it's also a space getting pretty crowded. At least that's my perception. Both the prebiotic and probiotic area is pretty busy. So I'd like to understand better, what is it that the uniqueness about you guys? And that's my question number 1. And question 2 is, it seems like you want to do many things at the same time. You're both going into pets, you have different concepts that you're building B2C, you're running clinical trials at Silkeborg University. Are you able to cope with all these activities?
Yeah. Okay, so on the first one, so prebiotics and probiotics have been known for a long time. A lot of that is snake oil, so there is a need for having science behind it, and that is actually what is happening now with the microbiome research. But in addition to prebiotics and probiotics, we also have a lot of bioactives coming from the plant material, coming from the seaweed material, but also bioactives which actually are created under the fermentation process. And we have a pretty good picture of that. For example, polyphenols coming up, lipids which are anti-inflammatory. And I really think this about the bioactive is what is differentiating our products to the other supplement products out there. So that's really where we see a huge benefit. And of course this very clear microbiome modulation that we also see is something which are very attractive. So the last question, so we have a lot of on our plates right now, and we can kind of code with it. We have very good support from the mother company, which have established a business with similar products with this, and I think that will help us out. But maybe at some stage we will have to take some decisions and say, okay, maybe we should create a third company on this. But right now we have good support from our mother company, and we can code with it. Of course, when the clinical data is coming in and we look into it, we will take some decision from that also.
So yeah, thank you, Søren, for a good presentation and very, very ambitious endeavor. So it's not 100% clear, but it is basically the same fermented product that you're applying in the different segments, the PET segment, the Crohn's disease trial, or is it different fermented products?
It is. So we have kind of developed a machine where we can use seaweed material and different plant materials. You could also change the bacteria which are used for the fermentation. So we're playing around with these parameters, and then we are dedicating products to each of these areas. But generally what we see is this microbiome modulation, and with a lot of the research the last couple of years on low-grade chronic inflammation, this is kind of what is the basis for our activities and the reason why we have chosen the different areas.
But doesn't that give you an almost infinite amount of variables to control, because you have different plant materials, different seaweeds, different bacterial length of fermentations, and how do you then come to what is best for the different indications? How do you select what is best for Crohn's disease?
Yeah, so on the Crohn's disease, that is actually historically from the compound or the product they have used at Silkeborg for nearly 3 years now, and that's why we have chosen that. But what we can see is, for example, it depends really on the seaweed material also you use, because some of the seaweed species, they are very antimicrobial, others are actually very anti-inflammatory. So in that way we have selected based on cell assays, other bioactive activity assays, what we should use.
Thank you very much.
Okay.
And now the last team from the BAA, Microbt, will be introduced to you by our entrepreneur in the house, Neil, again.
Okay, can I just ask a question? How many people in this room have your position, your career, based on making proteins or enzymes? Or use proteins or enzymes? Ah, okay, 40%. Okay, so for you, Microbt can help you out. It can help you make more of your protein, it can make your protein work better, and it can make it cheaper. So Microbt is a synthetic biology company with a range of technologies coming from the Novo Biosustain Center that can, at high speed, allow you to understand how proteins fold, to optimize the folding, to optimize expression, and to optimize the economics. And with that, I'm going to give you Maya.
Thanks a lot, Neil, for the introduction. I'm Maya. I'm the CTO of Microbt, and it's a pleasure to be here today and present the company to you. Why is that making noise? Microbt is a company that wants to establish itself in the protein production market. Protein production is a very major topic in biotech companies and pharma companies. And for those 60% of you that said that they are not involved into protein production, this is a protein. This is actually a very important protein. This is insulin. Insulin is creating the majority of revenues for one of Denmark's biggest companies, Novo Nordisk. And as we are here at an initiative of the Novo Nordisk Foundation, we should all be well aware of that. But also, on your everyday life, proteins cross your way. For example, in laundry detergent or in medication or supplements. Unfortunately, protein production is very often inefficient and therefore very expensive. And for several targets, protein production actually fails completely. Microbt has developed a technology platform that can help to make protein production more efficient and therefore cheaper, but the platform can also, yeah, I guess so.
You will get extra thing.
Well, I can just hold it and do like that.
Excellent, thanks.
Well, maybe not. No, it's working, it's fine. Yes, so Microb's technology can also enable the production of proteins that have previously been impossible to produce. The major bottlenecks in protein production are the expression of the protein, the folding of the protein, and the fermentation process. Microb's technology relies on three patented technologies, which can optimize expression, optimize the folding process, also enhance the stability of a protein, and optimize the fermentation process. By further developing those technologies and applying them, Microbt wants to establish itself in the protein production market and enable production of complex and difficult proteins. During our time here at the BII, we have accelerated Microbt from being an idea to a ready-to-go-to-market stage company. We have performed a market analysis where we figured out that our technology can target the biopharmaceutical industry as well as the industrial enzyme market. Both markets have around 40% of their products produced in prokaryotic organisms and can be targeted with our technology. Also, both markets are constantly growing, which is a good sign for us. We have also performed the competitor analysis in this market segment, and we have identified as our direct competitors companies such as Vectron Biosolutions, De Novo DNA, Expression Biotechnologies, and Engenes. All those companies have established themselves as protein production optimization companies. However, I will not go into detail here, but if you look at the coloring, you will see that each of those companies is covering a part of the protein production process. But Microb's technology is actually covering the whole protein production process. However, we have not yet established our technology for eukaryotic organisms, and we have also not involved scale-up fermentations into our portfolio. Two also very well-known players in the field are Ginkgo Bioworks and Zymergen. However, their approach and focus and also pricing model is very different to Microb's approach. So we do not see them as competitors and would rather see them as companies with whom we could collaborate. It is our unique value that you can optimize protein production without compromising folding of the protein. Our technology is highly adaptable. We can customize it for any prokaryotic expression host, any expression system, any promoter system. Our technology is coupled to a unique selection system which can enable the very fast generation of optimized strains and will therefore save your time. During our time here at BII, we have also developed a business strategy and a sales strategy. We have created a project plan for how to continue after BII and have calculated carefully a budget for that. And we found that with 3 optimization deals per year, Microbt will be able to be a commercially viable and cash-positive company within 2 years. Very importantly, we have also analyzed our freedom to operate and found that we are free to operate within all our 3 IP-protected technologies. During the time at BII, we also had the chance to write grant applications and to meet potential customers, but also like-minded companies. And here's a list of all the companies that we have been in contact with during our 10 weeks here. Last but not least, I also want to tell you something about the team behind Microopt. The technologies behind Microopt were developed at the Novo Nordisk Foundation Center for Biosustainability here in Copenhagen area, and at Stockholm University in the research laboratories of Alex Tofgard Nielsen, Daniel Daly, and Morten Nørholm. I myself, as the CTO, developed a part of Microopt's technology. The other part of Microopt's technology was developed by Jenny Landberg, who went through this exciting journey here at BII with me. We also have startup experience and industrial biotech experience, but most important, we have more than 30 years of academic and industrial expertise in protein production. And we are looking for an external board member to join our team, as well as a scientist to join, as a full-time employee, our company. To realize our project plan, we want to raise an investment about 5 to 10 million Danish crowns, and to initiate customer contracts, we want to expand our network into biopharma and industrial protein companies. And I hope that you can help us with that. And with that, I want to thank you very much for your attention.
Thank you, Maja. The very good presentation. And turn it off before. All right, thank you for a very good presentation. I appreciate that in spite of microphone troubles and everything else, you were bang on time, 7.0 minutes. Very impressive. So, and I also appreciate the 7 minutes didn't allow you to go into detail on exactly how your protein production technology is different. But maybe you can sort of tell a little bit about how you will go about introducing your technology in the value chain of other companies that produce proteins, because they will have to change their manufacturing, they will have to use your technology, and how big a barrier do you think that will be, and what's your thoughts on that?
Well, I don't think necessarily that a company needs to change their workflow if they integrate our technology. However, when it comes for pharma companies, for example, then of course we want to target like the lead optimization, lead development, because of course if you change something in GMP production, then it could be problematic. But our technology simply modifies the already existing expression strain. So it's not that we introduce a new expression system or
it's sort of an optimized expression strain that you're making. Okay.
Exactly.
Okay. Good. And the second question is you're currently asking for looking for 5 to 10 million, and what activities would that investment fund?
Well, first of all, we need to get hands-on scientists to do the research and a physical laboratory in office space. And then we want to further develop and integrate our 3 technologies. We already have tested parts of the technology commercially and had some success, but there is some development needed. And then we also want to go further to, besides doing business-to-business contracts with for other companies, we want to develop our own product line, because we think that our technology is extremely beneficial for difficult to express targets. And there are many of those targets on the market that have been shelved by companies or things like that, because they are very interesting but very difficult. And bringing those products to a stage where they can then, for example, by another company, be further developed and brought into market, that would be also our plan.
I just have one question, or maybe a suggestion. There's definitely a trend from corporates to externalize much more of their R&D, both to get the diversity and maybe different approaches to some of their R&D programs, but maybe also to make more of their cost variable. So maybe you want to put that as part of your offering to corporates. There's one thing I'm not sure I entirely understand. That's the business model that you have and how much money you can actually earn from being a service provider, and your reference to Ginkgo and Zymergen that you would more be a partner of theirs than a competitor. Maybe it's because I don't understand exactly what they do then, but to me, you're doing at least part of what they're doing.
Well, yes, there might be, for some parts, similarity of the approach, but however, Ginkgo and Zymergen are having those super high throughput automated strain engineering workflows, and what they do is that, as far as I understand, that they back engineer a strain first to the original one and then optimize that strain into the perfect expression host. This process, of course, takes a long amount of time, but our technology, as it is very targeted, only takes 3, maybe up to 6 months until we can deliver the optimized strain. So I think that there would be also, we have this folding sensing technology, which is very unique, and I think there would be like parts of technology, of our technology, that would be interesting for them to incorporate.
Let's give a hand to Maya. And also to all 6 BAA teams. Now the presentations of BAA are over, and soon you are just about to vote for the winner of Acceleration Award. And I would like to make a point that, yes, there will be one team that will go home with this trophy. However, we consider all our teams to be great winners. The progress they have made so far has amazed not only us, but also potential deal takers, and we are hoping to see outcomes of it. Having said that, very soon we will distribute this kind of voting cards. You can see all 6 teams and a check mark. You have to check one, and that should be the team, not the one that only impresses you the most, and not the one that is the furthest in the commercialization process, but first and foremost, the one that you believe has progressed the most over the program. We have taken teams in many different application scopes at many different stages, so it's simply not fair to say which one is the most investment ready, but it's all based on your gut feeling and the impression they have made. This is something what we would like to appreciate, people who are coachable, who are open, and who are driven. So listen to your hearts and decide who should get this trophy this time, and these people, they will be standing up on stage at the end of the program. Just a little bit on the timing. Now I will announce a break, and please start your break with voting so that we don't have cumulated votes at the end. I suggest that if we finish voting at 2, that's a right on wall, a fancy feature of our BII office. And then we meet here 2:10, so that we can proceed with the presentations from the incubation team. Is that a plan? See you soon.
Boom, boom. All right, guys, we start in a few minutes, so please take a seat. We have a few more seats over here, so if you want to get over here. Wow, okay, that was fast. Everybody got quiet. So now we move on to the next part, which is two presentations of our incubation company. So they all started in the beginning of September together with the 6 BAA teams you just saw. These guys are in the next phase of our of the BII program, which is the incubation part. They've all been granted 1.3 million euros. So these guys are here for a while. They will start working in the laboratories we just opened, which you will see a little bit later. The first presenter is Henrik. Henrik is from Retipharma, and I will let you introduce yourself.
Thank you.
Give him a hand.
Good afternoon. My name is Henrik Wissing, and I am the CEO of Retipharma. And in Retipharma, we do eyes, so that's the key message, if all. Our vision is to offer first-line treatment and protection against vision loss in degenerative eye disorders. It's about more than 250 million people on a global level suffer from one or more eye diseases, and for many of those, these lead to complete blindness, and which is extremely debilitating. Very early on, when we started, it was important to us to identify the indication we want to work on, because we are working on neuroprotection, and we looked at many indications, and we came to the conclusion that we are not going to treat all 250 million people on the first day. We're going to go on the other side. We're going to go on a rare disease, and we will look at protection at vision loss in retinal detachment. Retinal detachment is a disease where, as you know, the retina detaches from the inner surface of the eye, whereas you get ischemia, you lose blood to the retina. The only treatment today is ocular surgery. And if it's untreated, you will become completely blind after a number of days. And even with optimal surgery, more than 50% of these cases will result in substantial loss of vision. If you look at the numbers, I said it was kind of a rare disease. In the US and the EU, we have around 100,000 cases a year. When I say involving fovea, that is the subset we're going to look at. That's the serious cases where the fovea goes off the eye, and this is the case where you have a very high chance of losing vision. Years ago, people have calculated the cost of a loss of vision, and if you're in the working age, NICE has calculated that well, in 2004, the cost of for loss of vision is around 30,000 euro. We think if we can demonstrate proof of concept in retinal detachment, this will translate into clinical effect in other eye diseases such as retinal vein occlusion, diabetic edema, acute glaucoma, and further on. So this is a much larger indication, but we have chosen retinal detachment because this is where we think we can demonstrate proof of concept for a new indication and for a new treatment scheme that where we are kind of first on. A little bit on the candidate drug. We have a number of peptides. This is the first one, hence the number 001. It's a 33 amino acid peptide that is a selective NCAM ligand. This means it's a neural cell adhesion molecule it binds to. At the moment, we are looking at intravitreal, is eye injection, and the route of administration, which is kind of the typical way that the people are treated today for diabetic eye diseases. We have kind of a huge preclinical package for validation that this will have effect. We have shown it has neuroprotective effect in a porcine model of retinal ischemia. We have shown in a rat model of acute retinal ischemia. And at the moment we are at the stage where we are preparing for clinical tests, means we are starting up doing safety assessment, toxicology, and what we call CMC process for our protein. We think that if things go according to plan, we will be in patients end of next year, end of 2019. The standard care of treatment is that if you sit at home and certainly you have a curtain coming down in front of your eye, you go to the doctor and say, well, this is not very good, you have a retinal detachment, and we will submit you to the eye clinic. It's you will go with the typical for a few days with the detachment, and you will have surgery, and after up to 8 weeks post-surgery, you will have tests like visual acuity. Visual acuity is actually reading letters at the end of a table. You will also have something called test for nerve function, ERG and OCT, and I will come back to that. What we plan to do in our clinical study, and also as a treatment option, is that just after the diagnosis of retinal detachment, we will offer a pre-operative intravitreal injection of our peptide, thereby introducing a neuroprotective effect until the operation is finished. We will have readout clinical efficacy will be evident after 8 weeks, and there will be visual acuity. And as you can see, if you can read, well, maybe you cannot read the bottom of these letters. If you can't, maybe you have a problem. But when you come in before operation, let's say that you are able to read in the bottom end. So after the operation, many patients, they will only be able to read on the top 2. So the clinical outcome is non-invasive, it's very simple. You want to protect your eyesight and you will be able to read. And also mind you that if you can't read this after the operation, glasses will not help you. This is a loss of vision. It's your nerve cells that has been in apoptosis. Also, we will measure nerve cell function, something called ERG, I'm going to show that, and also nerve cell survival. A little bit on how we develop our drugs. The platform we have, what we call it the Retipharma translational platform for eye diseases. We test candidate drugs in animals, and we use endpoints that are identical to what is done in the clinic for patients. Typical, animal models have historically been difficult. They're difficult in predicting outcome in human diseases, but we have chosen a model where we use the same readouts. And here we have, well, this is from our web. We do not show animals on our website, so this is animal models. This actually, in this case, is a test tube. And there's 4 parameters. We use the same way of administering the drug, intraocular and topical drug delivery. We introduce ischemia to the eye by a chemical. So we mimic the ischemia that a patient is having in the retinal detachment. We use electron retinography, ERG, that also translate, and we use optical coherence tomography, OCT, that also translate. I have one data slide. I apologize for people who don't like data slide. It's a technical slide. So this is one of the key things we do in the company. We do ERG is a test that measures the electrical activity generated by the neural cells in the retina in response to a light stimulus. So you can see here you get a lens on the eye, you get a flash, and the amplitude, the height of the amplitude reflects how responsive your nerve cells are in the eye. So if you have your neural cells are damaged, the amplitude will go down. So this is the measurement we use in animal models to test the effect of the drugs, and it's the same that is used in the clinic at the hospital. A little bit about the company. We have an exclusive license from University of Copenhagen to IP on this family of peptide that has neuroprotective effect. We have, well, as you were told, we are kind of blessed that we got funded by the Novo Nordisk Foundation BioInnovation Institute with 1.3 million euros. So we are at the moment in pretty good shape. And this money is being used to prepare the drugs, as I said, for a clinical test, and we are now using the money for safety assessment, toxicology, and CMC. Of course, we are already now looking at what will go on after we have used the money from the BII, and we are now, of course, actively seeking partners and investors to support the clinical development program that we will start up by end of this year. And also we want to insource additional assets into our company with different mode of action. And we think that our front runner could reach the market somewhat maybe in 2023. I spent some time using the word we, and we are the board. We have Morten Albrechtsen, who is our chairman of the board, Jørgen Thorbal from Switzerland, Kasper Tin Hansen, and David Wolby. David Wolby is from the University of Copenhagen, the inventors of these peptides and also one of the founders of the company. We have Christian Klemp, medical director, also chief physicians at Rigshospitalets Glostrup Eye Clinic. It's always good to have a doctor in your company, and if some of you will happen to have a retinal detachment, he will probably be the one that will stitch it together for you. We have Andreas Sørensen, discovery science leader, also from University of Copenhagen, Anders Christiansen, clinical expert, Rigshospitalets Eye Clinic, Glostrup, Søren Emil Nør, Katja Saud, and Annika Runegaard are research scientists also working with us here. And by this, I thank you for your attention.
Thank you very much. And thank you, Henrik. I can take this one. All right, last presentation. We just need to get it's all the ones with the retinal displacement, so they're getting out to get an eye fix. What we'll have now is the last company, Stipe Therapeutics. We have Martin to present it, also known as the Sting peptide version of Elon Musk, I believe you told me. So no pressure. Martin, take the stage.
Thank you. I'll just have to go back once. So, good afternoon, everybody. My name is Martin Jacobsen. I'm the CEO of Stipe Therapeutics, a company with a first-in-class compound that can prime our host immune system to fight cancer. As we all know, cancer is a global epidemic. Increasing people are getting cancer, and we have difficulties to treat cancer, and it's also related to increased mortality. If we look at cancer, you can actually put it in 7 steps, as you see here to the left. So the whole idea with cancer is that during these 7 steps, our immune system is educated and primed to kill and seek out cancer cells throughout the whole body. However, this rarely occurs. One of the reasons for that is we have patients with tumors that are defined immune desert or immune excluded tumors. That is tumors where the immune cells lack functionality, is not present, or by means from the tumor itself suppress for its function. This means, we have a small group of patients which have inflamed tumors, which we also define hot, meaning we have immune cells that work, they kill the tumors, and this is actually the group of patients where we can treat today. You've heard about immunotherapy, chemotherapy, and radiation, and those kind of treatment mainly works in patients who have a hot tumor. So this leaves us with a major challenge. How do we treat around 60% of all patients with cancer to be able to respond to immunotherapy? We need new immune modulators. We need drugs that can make immune desert tumors and excluded tumors hot. So turning a cold tumor hot. That is what we can do in Stipe Therapeutics. Stipe Therapeutics is targeting a central compound of our innate immune system called STING. What we know is that activation of STING leads to a very robust and potent immune response in our body, and especially if you do that in the tumor, you can get a very strong inflamed response within the tumor. STING is a compound that my research group have worked on for the last 10 years. We know today that STING is activated by DNA damage responses or the accumulation of DNAs within our cells. It's activated by another protein called cGAS that produces a very small molecule called cGAM, which is a true ligand of STING activation. Activation of STING leads to very robust transcriptional expression and production of type 1 interferon and cytokines, which modulates our whole immune cells to become very efficient in killing cancer cells. Multiple biotech industries are now working on ways to activate STING. mainly by generating small molecule agonists that reassemble the CDNs, or actually using synthetic version of the cyclic denucleotides. However, we have to be careful about modulating the innate immune system. Systemic activation of stings is a severe risk factor, as you can actually produce a very strong cytokine storm that potentially could kill the patient. Therefore, the most of this company, especially led by Merck, GSK, and Aduro Biotech, they work with intratumoral delivery of sting agonists. So you only get a response within the tumor. What we have done in STIPE is we have developed a model that primes the host immune response. And what we use the endogenous sting activation that we know is occurring within the tumor. So tumors consistently express and release tumor DNA or induce DNA damage. And with our compound, we can prime this response. And therefore, we can also give it systemically. So what we discovered some years ago in my lab is that another host protein called I516 modulates the function of sting. More specifically, a very small area of I516 called the pyrene domain directly regulated the function of sting. So we developed small peptides mimicking this region, and we added a cell-penetrating motif to make the peptides go into the cells. We then looked in various immune cells, either with the peptide alone or in combination with a sting agonist. Here we used 2'3' And what we could see is that the peptide actually modulated the sting function. We actually primed the whole sting response to become more sensitized to DNA damage or CDNs. And this here is just an example. So far, the lab has screened more than 15 different compounds of peptides that mimic the function, and we mainly use a biological readout in terms of picking the right one, meaning which peptide induces the highest amount of type 1 interferon and cytokines. And this has generated a lead compound called ST101, which we know modulate the sting complex, but without eliciting an immune response itself. But when we put it together with a CDN or DNA damage response, we get a very potent immune response. We've moved this into animal models. We've used various models, metastatic melanoma, triple-negative breast cancer, colon rectal cancers. We have looked at the tumor growth in the mice and then treated with a peptide and looked at how it suppressed the tumors. And here we can see that with a high amount of peptide, we actually can get complete remission of the tumor. In colon rectal and 41, we get up to a 42% tumor growth inhibition with this model. And here we only use it as monotherapy, so we're actually exploring the endogenous sting activation. We then went on and looked at the function of ST101 in combination with a clinical grade CDN from Aduro Biotech called ADOS100. First of all, we see when we give systemic administration of ADOS100, we get a very strong level of cytotoxicity, which you can measure by decreasing growth of the mice, as well as inflammatory cytokines in the blood. However, when we did the same experiment with our peptide, we saw no effect on the growth of the mice, and we didn't see any systemic immune activation. So when we combine these two together in our animal models, what we could see is that if we use a low amount of the CDNs to prohibit systemic administration, we don't get a complete clearance of the tumors. But when we put it together with a peptide, we got a 100% remission, and as you can see all the way over here to the right, all mice that got the combination of peptide with ADOS100 survived the trial. So STIPE Therapeutics is quite differentiated from our competitors. We have a sting sensitizer, not a direct agonist, increasing the safety issues. We have the possibilities to do systemic administration, and we don't see any cytokine storm. We have a very strong and potent cell uptake and tumor tissue distribution. And we believe that using our peptide with current therapies, we can improve the efficacy of these therapies by modulating the sting response. So where are we going now with STIPE Therapeutics? We are in the process of lead peptide optimization. We're doing crystallization of the peptide with sting to improve peptomimics. And all of that should lead us to a preclinical selection. We are also working in our lab to expand the work on different immuno-oncology drugs with the peptide. And we are aiming to raise a seed investment of around $5 million end of next first quarter of 2019, which will enable us to identify and lead our preclinical candidate. So the company, as it is right now, consists of myself. I'm also a scientist at Aarhus University, and I co-founded this company with Claus Olsen, who's also a scientist at Aarhus University and involved in other companies in the past. We've identified a CMO and CDO, which will be announced early next year. We have a very strong board of directors. Some of you probably know John Haum and Magnus Persson. And we also have an additional member in the board that will be presented early next year. The company, as we see it, is an Aarhus-based company with a lab hub here at BII in Copenhagen. So we find ourselves as a Danish biotech company. Thank you.
Thank you, Martin. And let's see about that, who's the hub and so on, right?
Yeah.
No, but this is exactly what we want to show you. Scientists becoming entrepreneurs and bringing new science into the market for patients and the environment. So that was all the presentation. I hope you enjoyed it. Now to probably the most exciting part, which is the winner.
Which is me. First of all, first rule of our office space is to leave the room in exactly the same shape as it was when you came in. So, for the pictures, nobody wants to have my sketches on the background. And now the most exciting part of today's early afternoon, which is announcement of the winner. And for this, I will bring for you our amazing panel, Sebastian, Stefan, and Soren, to announce the winners. Please come up on stage.
All right. Well, thank you, Monica. And we didn't get a little envelope, although I guess we know the winner already. There is the envelope, and Thomas will now eat it. No, I think on behalf of the panel here, we're all impressed. These were 6 very different but very good projects, good pitches. They were all limited to this 7-minute format, which is super difficult. I think they've all learned a lot by being here in the BII and learn how to come to the core and the most important point of their projects. So very difficult. I think there's been a lot of voting going on. There's been the online votes. There's been the hidden votes. There's been the engineered ballot box and all the other things that has now been counted. The Russian votes, yes. So happy to announce the winner of the 100,000 BioDollars. And again, they're called BII dollars, BioDollars, very important. When you see deals announced, right, they're always bio company X make a deal with pharma company Y. It is for $8 billion, typically $2 million up front, and then the 7.999 billion are BioDollars, right? So they may be paid at some future point if certain milestones are met. And this is the case here. We are also operating with BioDollars called BII dollars, 100,000 of those. And with that, I think we cannot keep the waiting any longer. We have to announce that the winner is Circle.
Congrats.
Yeah.
Congrats. Congrats. You have the prize? Yeah. All right.
Oh my goodness. Go ahead.
Yeah. All righty. You keep it. Let's give them a hand.
Speech, man.
Yeah,
you're the official.
Yeah, thank you very much. I don't know what to say, but we're happy to win. Yeah.
Anyone you want to thank?
We want to thank people that voted for us. They're the most wonderful person on this earth. Thank you very much. And we're going to make you proud. We really want to make an impact and help women and couples, and so we're going to give it 100%.
Thank you.
Yeah, thank you. Just before we end here, Søren, any remarks from you sitting in our panel?
Excellent presentations, and for me, all 6 of them are excellent examples of what I would like to see much more of the coming years from the BII. I think we need much more of this, and of course, on behalf of the university, is extremely happy that most of them, or all of them, are of course science-based. So having that also as an impulse and a positive impact back to the universities, so much more of our research will actually come to life out in the real world, so to speak. So excellent examples of that. And a small reflection on all 6 of them. I'm really looking forward also to see that each of you become, this could sound critical, a little critical, but to be a little bit more interdisciplinarity in your focus, maybe entering more and more different competences than the discipline you actually origin from. And I think that will also be in the spirit of the BII we're going to see to develop in the coming years, not just to you, but also, to the other teams. But congratulations to you, but also to the rest of the teams. Excellent presentations and good products.
Thank you so much, Søren. Sebastian, any reflections just in closing?
Well, I also think definitely that the presentations were really great. But I would like to say a big thank you to the entrepreneurs and praise them for how brave they are. They're leaving secure jobs to pursue a dream, and because they want to have an impact. And this is something we just need in Denmark and in our ecosystem. It's not in everyone's DNA to do this, so they're really, really brave. And hopefully there'll be 2, 3, 4 success stories within the next 24 months. And I'm sure there's going to be a cascade of more young people with skilled professionals from the universities and the business that want to join them and hopefully create a lot of new companies for Denmark. As a corporate representative, I can also say that we have a lot of ideas that are not being matured in our companies today because the risk profile is maybe not the right for a listed company. Or sometimes it's simply just because it's not our business. But still the ideas are there, the talent are there. And I think what the foundation has enabled here is the first step in an ecosystem that is needed in Denmark and in our region. So big thanks to all the entrepreneurs. Thank you.
Right. Stefan, as the
raisin in the sausage.
Yeah, well, and the seed investor, the man with the money.
Well, okay. Well, I can only second or repeat all the good words. Very, very impressive presentations, and it's been very good to work with BII. I think we look forward to our collaboration going forward from Novo Seeds. Many of the projects that we're presenting here, of course, are projects that we know, and we look forward to keep it that way, to have a good interaction on projects we know, that there will be projects that you work with that we don't know, and where hopefully all the efforts here will make many more interesting investment opportunities available. And we look forward to invest in those promising companies as they develop in the coming years. So great initiative, great start. Thank you all. And thank you to the BII team.
Thank you so much, and thank you to the committee or the panel for helping us out. I think actually we need to do one thing, because this is a competitive effort, of course, what we are doing, and we have talked much about the sound Darwinism in the selection criteria going on in BII and in the promotion of. So we have some winners, but we also do have 5 other teams that I would actually like to have here on stage for a group picture. So please, all the BIA teams, join us on stage and give them a big hand as they arrive on the stage. So as they would say, it's time for picture taking, but we need yeah. Alrighty. So we're all here. Ladies and gentlemen, this is the very, very first batch of the BAA demo day companies and the first batch of our trial run of the BAA program. So a last round of applause for these wonderful projects. And all of them are, in their very own way, winners. With that, I would like to thank you all for coming for the demo day. We are now going to, as we are kind hosts here, ask you to leave because we need to clean up the room as we have an opening event going on in just 15 minutes or so. But as you move towards the end of the room, you can either go down the stairs or you can drift into what we call the square, where the opening ceremony will commence in just a few minutes. Thank you very much for coming and enjoy the rest of the day.